All right, so we're gonna do a classification algorithm today using a logistic regression model. I put a summary here about this activity. In this activity, the goal is to generate a model that will be able to classify a neuroblastoma patient as having high risk or low risk disease. This prediction is going to be based on the gene expression pattern of a tumor from the patient. The hypothesis that I'm using here is that by creating a model, we can estimate the risk category that a patient belongs to with a high accuracy, and we're going to discuss accuracy in a little bit, using these 22 predictor variables. And that last point there is just the references for this notebook. Now I want to ask you, do you guys think that this can possibly be useful for anything, and what is this going to be useful for, based on the very great nice presentation that Anna gave us last Wednesday? What do you guys think that classification could be useful for? Rishima, why do you think, or do you think that this could be useful at all?
Sorry, you cut out. Can you hear me? Like you're stuck for me.
I can hear you. Can people hear me or no?
Hello?
We can hear.
Does anyone hear me? Nobody hears me?
We can hear you.
Yes, we can hear you.
Oh,
Yeah, I can hear you.
Okay. Okay, Eric, do you think this is useful, and why do you think this can be useful?
I imagine it's useful because you could take you could take a lot of data and then apply it to specifics of a patient. Like if I understand how it works, and then being able to match if there are any similarities between like other high risk or low risk patients and then the new patient. Or I might not have the best understanding of it. I'm not sure.
No, I think your answer is in the right path. Are you saying that we can get to analyze a bunch of tumor data in some patients and then use that information to get to know about the risk level of a new patient? Is that what you said?
Yeah, that's what I'm trying to say. Yeah.
Okay, yeah, that's what this does. So that's exactly what it is. We are going to generate a model that knows some information about what defines a high risk patient or a high risk state for the patient. And then when we have a new person, actually, it should not have to be a neuroblastoma patient. If we have a person in that doesn't have a neuroblastoma, the model should be able to know whether the person is a high risk, can be classified as a high risk patient or a low risk patient. And so I would say also that this is important because let's imagine that we have a patient and we give them yes, Victor, it's related to what the guest speaker talked about in the last class, but in the sense that we can create a model that is going to understand what one class is versus another class, or one label versus another label. So when I say label, I am talking about the classification that a sample has. In this case, well, for this case here, we have high risk neuroblastoma or low risk neuroblastoma. So what I was trying to also mention is that once we know what characterizes a high risk state, imagine that we can give a patient a therapy. We can give them some treatment, and we can also quantify what is a high risk after the patient receives the treatment. So we have the patient in state 1 and the patient in state 2. In state 1, we can estimate how much risk they have or which risk category they belong to. And in state 2, we can also quantify how much risk they have and which risk category they belong to. So in other words, in a very simple manner, first, what we're going to learn right now is going to allow us to classify a patient into high risk or low risk. And with that, the way that this is designed, we can also in case number 2, we can quantify the risk that a patient has after therapy. So if the therapy we gave the patient was able to change their risk category, this is an algorithm that can quantify that, or that can classify how much risk the patient has after receiving a certain therapy. Okay, just revising what we talked about in the last lecture, supervised and unsupervised methods. Today we are talking about classification. This is a classification machine learning method. And the other method that Anna talked about in the last class was clustering. So classification basically relies on labels. In this example here, you can see a scatter plot of the height versus weight. And we can see that the trend here is that children have lower heights and lower weights compared to adults who have higher heights and higher weights. The difference between classification and clustering in this case is that we know, or the algorithm knows, what an adult is, which data set comes from an adult data set. But in the clustering, because it's unsupervised, right? It's an unsupervised method. We use metrics to differentiate clusters 1 and 2. So which sort of metric could you use to differentiate clusters 1 or 2? We could use, for instance, a metric of proximity, right? So one very common method that is used in an analysis like this is correlation, getting to know a correlation between each one of these triangles here. We could use this information to separate these elements or these samples from these samples. Because in the clustering, what we can do is we can get to know the elements that correlate more or less. And if they correlate more or less, then they tend to be clustered together. And the reason why I'm using this metric, or the reason why I am stipulating this for this data, is because I'm interested in knowing the relationships that are happening in this scatter plot so that I can develop some sort of explanation to why do these samples cluster together? Why are they together? Remember in the unsupervised algorithm, we do not know that these triangles are from adults, right? We do not know that these are from adults. And clustering, therefore, is a method that we can use to group samples together. Okay, so exactly this type of information was used to detect subpopulation in neuroblastoma that drives aggressiveness. So isolation and study of these neuroblastoma cells has allowed the study of phenotypes that drive aggressiveness. So as I mentioned, we have two main cell populations in neuroblastoma. The mesenchymal cells are rare and aggressive. They have stem cell-like characteristics, whereas the adrenergic cells are more common and they are more treatable than the mesenchymal cells. Hierarchical clustering is one of the machine learning methods that have been used to identify the pattern of gene expression of adrenergic and mesenchymal cells. So clustering is a segmentation technique that partitions the data into several groups based on their similarity, as I was saying. It's a unsupervised method. It uses data that is not pre-labeled. The data is not labeled. Here are some algorithms for clustering, including hierarchical clustering. So let's now discuss a little bit about the unsupervised method that was used in neuroblastoma research. Do you guys have comments or questions about any of these informations that I mentioned here before we move on to hierarchical clustering in neuroblastoma? Okay, clustering can be used to separate the genetic profiles of cancer cells that drive progression or defeat the disease. Neuroblastoma cells are differentiated. They are similar to neurons. Mesenchymal cells are non-differentiated cells. You can also say that they are de-differentiated, and they are thought to behave like stem cells. Mesenchymal cells are resistant to chemotherapy. That's why they are so dangerous. So the technique that people use it to differentiate these types of cells, well, first of all, we always do protein expression blots, right? These blots, they measure the expression of a protein. And so you can see in these blots, for example, for example, FOX2A and FOX2B are adrenergic markers. That's the reason why you see FOX2A and FOX2B being expressed in adrenergic cells, being more expressed in this other adrenergic cell, and more expressed in this other adrenergic cell pair than the mesenchymal component of the pair. By the way, in adrenergic, I told you that adrenergic cells and mesenchymal cells, they can interconvert, even though they have the same genetic backgrounds. That's why we call these cells, these are called, for example, Shep cells. These are isogenic cells, which means they have the same genes or the same genetic, but they differ on the pattern of expression of proteins. You can see that these adrenergic cells, they have higher expression of adrenergic markers, whereas mesenchymal cells have low expression of adrenergic markers. Okay, so this was for the proteins, and this technique is, it's older. This technique is older, much older than this next generation sequencing technique. It's a it's based on the next generation sequencing, and what we see here as well is that there is a very characteristic separation of each line. This is I was trying to remember that this is an RNA-seq experiment, and in this RNA-seq experiment, we can also realize that the pattern of gene expression is very characteristic in the adrenergic cells and very characteristic in the mesenchymal cells. So you can see that a number of genes have higher expression. You can see that this is higher expression from the score here, more red, higher expression of adrenergic genes in the adrenergic cells, and higher expression of mesenchymal genes in mesenchymal cells. This state of differentiation has led to scientists willing to find an explanation for these states of protein expression, these states of gene expression. One of the explanations is the idea of super enhancers. And so super enhancers are regions of the chromatin that are maintained open to facilitate the expression of proteins that keep the differentiation state of the neuroblastoma cells. And by keeping the state of differentiation, what we mean by that is that so there is high expression of the adrenergic genes here, right? And in the adrenergic cells. And the reason for that is because the chromatin, the DNA, is organized such that it maintains a high level of expression of those proteins. And that is very tied to the idea and the concept of a super enhancer, because in this figure, you can see that there's a lot of transcription factors bound to a super enhancer region. That a super enhancer is a combination of enhancers that come together. And because they come together, this can attract the cellular machinery that is responsible for the expression of the gene that is just downstream of the promoter region. So you have the formation of a cellular machinery that promotes the synthesis or the expression of the genes that are characteristic of the state that the super enhancers represent. And in this case that I'm just pointing here, this is the adrenergic state, right? So in the adrenergic cells, there's going to be only the expression of adrenergic markers. There's going to be very low expression of mesenchymal markers. And remember, the adrenergic cells are differentiated and they can be killed by chemotherapy. The mesenchymal cells are not differentiated. They are less killed, or they cannot be killed by chemotherapy. And that's what explains the higher expression of you can also call this as a pattern of differentiation, right? The cells maintain a high expression of those genes because it needs to maintain its state as an adrenergic cell in this case. So you can see here that this machinery and the coming together of the transcription factors makes it so that there is a lot of transcription factor molecules in the region, and this can recruit more and more of the cellular machinery that is responsible for the expression of the genes that this has to express. These would be algorithmic steps to identify super enhancers. First of all, there has to be a high enrichment for chromatin immunoprecipitation. It's an experiment that can help us pull down regions that are bound by some transcription factors, okay? And then we can use a threshold number to calculate the amount of enrichment of the amount of enrichment for the binding of the transcription factor to the genomic region. And with that, we can, as I said, we can establish a threshold where we are going to be able to identify the super enhancers. So if there is a lot of binding of this MED1 is just one example, and I put the definition of the MED1. It's mediator of RNA. It's mediator of RNA polymerase expression. It's this protein here. So if there is a lot of binding of MED1 to that region, it means that that region is composed of a lot of enhancer sites, which consists of a super enhancer. This has led all of this, both of these information here and this information and the algorithm that we can generate out of this has led to the idea that there are super enhancer regions in the neuroblastoma cells that separates the epigenetic patterns of the mesenchymal cells from the adrenergic cells. You can see that the enrichment of the super enhancers are higher here for the mesenchymal cells and higher here for the adrenergic cells. And that what that means is that we are able to discover a pattern that can differentiate, a pattern of an epigenetic pattern that can differentiate an adrenergic cell from a mesenchymal cell. Now that we know how we can classify a mesenchymal cell and an adrenergic cell, let's develop an algorithm to be able to classify not a cancer cell, but let's develop an algorithm to classify a tumor, a patient, right? Let's develop an idea to classify a patient. So the foundations of the linear regression model. We are going to use a probability function to calculate probabilities of a patient belonging to a class or the other. And what does anyone know which classes we are talking about here? We are talking about the high risk class, right? We are going to calculate whether whether a patient belongs to the high risk class or the low risk class. And the way that the method, the maximum likelihood estimation method does it is it calculates slopes here. And so by calculating these slopes, it's able to tell whether a patient falls more into the yes region or the no region. And the probability function that we are going to use is a probability function that is similar to a line, right? Remember this coefficient plus x times this other coefficient is what we define as a line, And this is basically an equation that we use to calculate a probability. This probability is in the logarithmic scale. That's the reason why it's called a logistic regression, okay? Because it uses a log of the probability of the variable that we want to identify. So here, you're going to see that each feature or each column or each variable is a phenotype expression that is given by the gene expression of the phenotype that we're trying to study. And here basically I'm talking about the columns in the data frame that we studied in the past lectures. So in reality, the model for a probability that this algorithm that we're going to use is going to calculate, it is going to be an equation that looks something like this. The probability can be calculated as a constant plus a coefficient that multiplies the variable, the expression that we quantified, okay? Remember the hallmark hypoxia? We quantified how much hallmark hypoxia each patient had. Remember the hallmark inflammatory response? We also quantified how much hallmark inflammatory response each patient has. And I told you in the beginning that I actually chose 22 of those variables. I think that the hallmark hypoxia is actually not among those variables, but there are 22 of them. And what those 22 variables are measuring, they are measuring some phenotypes that we are interested in the neuroblastoma data set. I hope this makes sense. This figure is from an example that is from the tutorial that I'm following here. But basically what this means is that the classification, to classify a user or a patient, whether they are going to buy something or whether they are going to belong to the high risk category is going to depend on a number of variables. And I just told you that these variables are the columns that we studied in the past lectures. Does this make some sense? Does this make sense? Do people have comments or questions about this?
Yes, I have one.
Mm-hmm.
Can we use, or I mean, can we have a tiered outcome variable?
Yes.
I mean, beside yes or no, or
Yes.
in
Yeah.
the example, high risks and low risks could be median risks, a tiered outcome variable.
Mm-hmm. Yes, we can do that.
Thanks.
Mm-hmm. All right. And, when it comes to showing the organization of the data frame, you remember that the high risk category is yes or no. Maybe just the way that we have it here, we can have yes, no, and maybe there. I think that it would be able to differentiate, but then if it throws some error at us, we would have to troubleshoot that. All right, so let's get started here. The first step is going to be uploading and processing the data. And for that, we're going to go back here to the drive, the shared drive. We can go to data, and it's going to be this object here that we're going to download. Let's go ahead and download this object. I hope I downloaded it to my desktop. No, it's asking me now. I'm going to download it in my desktop. It's here. I'm going to go ahead and open our studio. I guess at this point, I do not need to use the console with you guys. So I'm going to just copy this and run these libraries. It went okay, and I'm going to run this command. I need to modify the path here so that it looks for the data set in my desktop. And so here I can come to the end and move on all the letters and hit tab so that it auto-completes. So I do not have the path problem that one of you were describing earlier. So I can hit enter. And now if I list, I see that I have that object there. We can count the number of dimensions. So interesting, this object is much smaller than previously. We still have the 498 lines, but only 26 columns. So I'm gonna use the view function here to view the, by the way, another name that I call that data frame, it's a Kozak data set. That's why I'm calling it Kozak, but it's the same data set, okay, guys? Let's take a look at this data frame. So the first column that we have here is the sample ID column, and then there's McCann expression, there's STC1, P4HA1, and so on and so forth. I see HIF1 is the hypoxia-inducible factor. I see T-cell exhaustion. I see the hallmark inflammatory response. Oh, hallmark hypoxia is here. I was wrong. It is here. There's also hallmark TNF alpha signaling, and so on and so forth. And we have high risk. We have INSS stage. Jose, by the way, this is a more than two outcomes possible. I think it would be interesting to take a look at to understand if the algorithm can use this variable instead, but we have to do some modifications to do that. And also here I have the McCann status. Remember that these are the clinical variables that we discussed a while ago. These are variables that tell a lot about the high risk category that that a patient can belong to. And so you guys know that McCann is a variable that has a very high impact on the high risk. Remember, the high risk patients have a very high McCann expression values, right? Maybe you remember seeing that plot. So I was expecting that McCann, so the one in the right was McCann status, but the one in the left is the McCann expression. And to my surprise, this did not give a very high weight for the McCann, and I do not know why that was. But anyways, let me go back here. So I'm going to remove the problematic columns. For some reason, I have to remove the sample ID column. Let's just go ahead and copy that and paste it here. And now, because I removed the sample ID, now I do not have that as the first column. Now I have McCann as the first column there. I need to make high risk, INSS stage, and MIC-IN status as factors. And I'm going to make MIC-IN numeric now. I'm going to change the name of the variable, but I do not know why it's necessary. Is everyone, are you guys able to follow? Is anyone having any problem to follow along so far? Alexandra, are you able to follow?
Yeah.
Okay. Okay, Victor said that he's also able to follow so far. So what I did was I just changed the variable name. And now I'm going to remove I'm actually going to remove the INSS stage and MIC-IN status information, because what we are doing is we're classifying based on the high risk, right? So now that we have, let's take a look at the Kozak underscore DF. We have, it's telling me that they are 23 columns in total. And you can see that the high risk column, can anyone tell a difference between high risk and the other variables? Eric, can you tell if there is any difference between the high risk variable and the other variables?
Is it that there are only two states and that the variables are characters and not numbers?
Yes, they are characters, they are not numbers. And these characters here, they represent the labels. They represent, and the labels here means what I want to determine if a patient that I do not know is or is not. So in the future, I want to basically be able to understand if a patient is high risk or low risk based on the variables that I can quantify, okay? And so when I say that I want to classify a patient in the future, what I'm basically saying is that I want to be able to tell if a patient belongs to one of these two categories in this case. So now let's go ahead and start doing that by creating the model. And here we are going to, remember that Anna said in the last lecture that we need to separate two groups. One to be the train set and one to be the test set. I am calling them the train data frame and the test data frame. So in the train data frame, I'm going to have R look at the high-risk patients and the low-risk patients and understand what pattern exists there that makes them high risk and which patterns make them low risk. And the R is basically going to understand that by applying this logistic regression model that I just touched upon. And it is by calculating the probability the way that I showed with the maximum likelihood estimate, the MLE calculation that I showed above. So I want to repeat the hypothesis here so that it's kind of clear for us, so that we have that in mind. And so the hypothesis is basically what I want to do. okay, what I want to do with the data. And another reason why I'm putting the hypothesis statement here is because I want to share with you guys that once you interact with the programming language and with the computer, you can basically think of a lot of things that the computer can do for us. We just need to understand how we are going to ask our questions, the way that the the way that the computational language is going to understand, and the way that the machine is going to understand the command that we are asking, because basically we can calculate. I'm not going to say we can do anything, but I'm going to say we can do a lot of things. We can get answers to a lot of questions if we ask the questions the correct way. And one way of putting the questions that we have is by writing a hypothesis statement. And so our hypothesis here is that by creating a model, we will estimate the risk category a patient belongs to with elevated accuracy using the 22 predictor variables. Okay, we are going to be able to predict using some measurement of accuracy using these 22 variables. So we are going to split the data, and in order to split the data, we are going to generate the splitting. Sorry, I need to go one step back. In order to split the data, we need to choose which patients are going to go into the train set and the test set, right? And the way that we do that is we use this initial split function. And in order for R to be able to split the data frame randomly, I'm going to use this initial split function, and I'm going to specify the proportion of samples, or the proportion of patients, the proportion of the 498 patients that I want in the train data set. And in this case, I want 80% of the patients in the train set. Well, I would think with myself, how can I make sure that I'm always going to have high risk patients and low risk patients in the train set and in the test set? I am not entirely sure about the answer to that question, but what I can tell you is that that's going to be one way to explain this set seed command, because what the initial underscore split function is going to do is it's going to randomly choose which patients go to the train and which patients go to the test data frames. Okay. So in order that random condition is reproducible, in other words, in order for that random splitting to be able to be repeated later, I need to tell R how to generate the random splitting. And that has to do with the idea that we have to provide a seed generation, which means we have to we have to tell R a number to start with, because otherwise that number is going to be what I am calling here pure random. Okay, if I do not tell R a number to begin with, to start with, it's going to think of it by itself, if I can say that, and then I will not be able to reproduce the patients that go into the train set and the patients that go into the test set. So in answering my first question, I cannot guarantee that all the time I'm going to have high risk patients and low risk patients in the trained data set, but I can guarantee that if I can start, if I can run the initial split command such that it splits the patients in the train set with high risk and low risk, and in the test set high risk and low risk, I am going to be able to repeat that later. So that's what this line does. And now we can construct one. And by the way, I did not run the seed set here. So I'm gonna do that now. And I'm gonna copy this line and run it now. And now we can construct the train set. And I can construct the test set. And let me see here. Let's now ask R which class the train data set belongs to. It's telling me it's a data frame. So I can, well, one thing that I know so far is that anything that is called a data frame, I can, no, sorry, view train. Anything that has class data frame, I can apply the view function to, okay? So I'm gonna hit enter now. This is what the train set looks like. See that it has 397 lines and still 23 columns, just like the COSAC data set. That's the original data set. But remember that here we have 80% of the data in the train set. And the test, I'm actually going, okay. And I'm going to ask what class has test. Oh no, class. It's also a data frame, and here it is, the data frame. It has 100 lines, and 20% of almost 500 is 100, so it makes sense that it has 20% of the data, because 80% of the data is being used to construct the trained data set. Are you guys able to follow along? Alexandra said yes. Advaita said yes, true. Great. Okay. So as I said, the trained data set receives high risk and non-high risk patients. So the data is trained using high risk and non-high risk labels. Let's see if we can see that here. Where's the... I can see it here, but I do not know why I cannot see it here. Or maybe okay, I can see the high risk column here. I only see no's here, but if I click 2, 3, 4, maybe 40. Yes, in the 40 that is yes, 38, 35, 33, 31. There are yes and no high risk patients. So definitely I can see that there are high risk and non-high risk patients in the trained data set. So the model is going to take a look at what a high-risk patient is and what a non-high-risk patient is. We are now going to do a step that is called fitting and evaluating the model. In order to do that, we're going to, if you haven't installed the glmnet package, you can go ahead and install it using this command now. I already have it installed. So let me just see if library glmnet. I just loaded it here. You can load it as well. Were you guys able to install it? This is the syntax to install. Okay. So this next command allows us to train the logistic regression model. We can read information about this logistic regression function. I put the reference below. If you scroll down, you're going to see this reference here. And there's also information about this mixture and penalty parameters in this other reference. For example, so we are using, this is called generalized linear modeling, the GLM net. Note that we are using the classification mode because we want to do a classification. We want to be able to classify high-risk patients in the train data set. And we want to use a logistic regression model with these parameters. So let's go ahead and copy this here. So we generated our model. Let's see what R tells us about the model. Let's ask what class it has. It's a model fit, and it's also a lognet. We can now take a look at what the model looks like. If I look at the object itself, it tells me it's a parsnip model object, and then it tells me what the call was, and it shows me this object with a df percentage dev and lambda columns. I can also take a look at the STR of the model, the structure of the model. This is what it looks like. It tells me it's a list of 6. It has 2 levels, yes and no, which makes sense. Spec, I do not know what it is. Penalty, I know that I gave it that parameter. Oh, 0. I gave the parameter 11. It tells me it has 0 here. Let me see if there's anything else that I recognize. I see here the mode as classification. We set the mode here as classification. There's not a lot here that makes a lot of sense to me. I can see here in this line that we have some of the gene expressions of the 22 features. in this line here, terms, it's interesting because it seems like what this is doing is it's calculating high risk using these predictors. And this seems to be the equation that model summary. It seems to be this equation. Okay, I'm just going to go back here. And so let's run this command here. So we have the term, that's basically the predictor, and it gives us an estimation of the importance of the coefficient of that predictor to the probability that it's calculating. So it's able to estimate how much each predictor contributes to the model, basically. Now let's come here to the predictions. And so the command to predict the class, that means the command to predict whether it's high risk or low risk is this command. So we use the function predict, and then the model that we constructed, and this parameter, new data. Remember that we constructed the model using the trained data set. Now we are going to use the test data frame. Remember so this is the train, this is the test. And remember that the train had almost 400. That is 397. The test has almost 100. Okay, so let's go ahead and run this. I can ask R what pred class is. It says it's a data frame, so we can view it. So the predicted class, I think this is very interesting because what this is telling us is that R was able to generate a model, and we can, my goal is to be able to understand everything that R used to be able to, based on the 22 predictors that I gave it, it used them to predict whether this person was high risk or not, and it told me that it was high risk. This person was high risk. This person was not high risk. And this was not, and so on and so forth. So I'm really interested in understanding how the model and how R was able to tell me or predict to me whether a person is high risk or not high risk. And one way that we have to estimate in which percentage of the time that prediction is correct or not is calculating something called the accuracy. So I am interested in understanding how R can tell us whether a person is from the high risk class or the non-high risk class. I'm also interested in knowing if R can do this accurately. And one of the reasons why I'm so interested in this is because I want to be able to give a patient a therapy and have a machine tell me whether the therapy increased or decreased their disease risk. Because if we can prove that a therapy can decrease a risk, and we can prove that that is something that is significant, then we have used our computational skills to do something good, right? And so those are reasons why I think this is interesting. Okay, so we were able to generate a prediction with just 3 lines of code. This is great. And now let's take a look at the probabilities that are used to assign a label to the patients. That is by calculating the predicted probability instead of the predicted class, right? So let's take a look here. The predicted probability. We can also view the predicted probability. So for this patient, the predicted probability was 90% that the patient is high risk. Is there a way to compare the results in pre-class to high risk values in the test data set? Yes, that we're going to do that in the downstream. And that's the reason why we want to do that is to calculate the accuracy. So what I want to do now is hopefully the 1, 2, 3, blah, blah, blah matches 1, 2, 3 here, but we will know from the probability values, right? So, so for these 2 first patients here, I would say that it would be yes, because of the probability values. And it makes sense because here is only 7 and 9, almost 10% probability that these patients are yes. So it's a very low, so they are no. And so we can see that it's a no for all patients below the number 2 there. No, 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 because it's very low for all of them. Yeah, it's making sense. So I am inclined to say that this patient number 1 is the same here and here. Yeah, I'm inclined to say that, although I'm not 100% confident yet. But let's continue here. Now we come to the part that Eric was just referring to. We want now to evaluate the model. And in order to do that, we have to consider the accuracy, the formula to calculate the accuracy. And that formula is correct classifications divided by the total number of classifications. In order to do that, we have to build the data frame results. And it's important to explain the steps in this way. We're going to construct the data frame results by selecting the high-risk column in the test data set, because if you remember how we constructed the test data set, these are true labels, correct? These are real labels. The high risk column here is real. We know that this patient is high risk, this patient is not high risk. So the first step is we select the high risk column in this test data frame. As I said, they are the real labels. And what we're doing next is we are binding the predicted class and the predicted probability. So I'm still thinking that we're assuming that the that the order is not being changed. I'm still assuming, I am sorry that I cannot say I am 100% sure that the order is not being changed, but I cannot. So I'm just being transparent, I guess. But the order is not being changed. I'm assuming the order is not being changed. So again, we are constructing the results in two steps. The step one, we select the columns in the test data frame, and then we bind to the predicted class and the predicted probability. And this is what the results data set is going to look like. You can see here that, well, at least for these two, for the lines that we see here in the test data frame, the predictions are correct. The class prediction is correct. And so now we can calculate the accuracy of the model using the accuracy function. And if we do that, we will see that the model has predicted 91% of the time that a person is high risk, or is of the high risk or the low risk category, or non-high risk category. I'm just trying to remind myself of what the accuracy formula is. Yeah, correct classifications versus the total number of classifications. I would have to break down the correct classifications, because the correct classifications account for the high risk predictions and the low risk predictions, right? Well, as far as I can tell you, everything so far here is accurate. So this should be well, what I'm saying is that we can trust this number. So the algorithm, using everything that we used so far, it is capable of predicting the correct label in 91% of the cases. 91% of the time it can predict correctly. We have 7 minutes left. I want to ask you guys if you have questions or comments, and most importantly, I guess, if this makes sense. You do not have to understand all of the steps right now, but according to following the original tutorial, I did the steps correctly, and with the steps done as they are right now, I can say that 91% of the time the algorithm, the model is predicting the correct label. Do you guys have questions or comments or concerns about how we did this? Yes, Jose, go ahead.
Yes, I can comprehend a little of the logic behind the algorithm or model, but I think when I have to make my own model, could be a lot of bit difficult,
That's fine.
because I think it could be complicated.
You think it's complicated?
Yes.
Yes, it is
Uh,
complicated. It is complicated, but I think it's from the perspective that this can be used to basically diagnose a patient, right? Ultimately, ideally, this can be used to diagnose a patient with high-risk neuroblastoma. And number two, with this, we can understand if a therapy increases or if a therapy can change the risk category of a person. I think I think it's fun trying it, and it's fun the what we can gain out of it. I think it's worth learning it. Yes, Eric.
For this kind of use of machine learning, is there kind of like a standard where like let's say the estimate, like is there a standard where above a certain percent of like accuracy the model is more viable compared to, like, let's say if it was like 80%, that like algorithms with a 80% accuracy or above are usually accepted, or
Well,
anything below it isn't accepted?
I would say that for diagnosing, 91% is pretty low. Because what I am showing you here, we did not consider so much, for example, how the data was generated, right? How the counts, how the gene expression was quantified, if it was normalized or not, which sequencing method was used. And the reason why I'm bringing that is because in order to build a tool or build a machine that is going to uh, I do not want to say that we are going to rely diagnosis only on this, but in other clinical tools that, for example, the MDs, the medical doctors are going to have. My understanding right now is that we must increase the accuracy to more than 99%. But for this method, for example, I do not think that I am at the stage where I am willing to have a 99%. I, I do not think we are at the stage where we can expect this to have 99% of accuracy or so. But I still think that 91% for the step that we are at, for the stage that we are at, is pretty high. I would so with this number, 91% is a high number. I think in general, machine learning methods, good machine learning methods are going to be considered good if they are something above 70%, is what I read in one of the papers. But I think that for the medical field, we expect a much more higher accuracy than that. Jose?
Yes, I have a question relation. Okay. Um, for you, what would be the minimum value in this case for the accuracy of prediction?
No, I think at least 90% right now should be fine. Is fine. At least 90%.
Okay.
Yeah. And there's always other variables around, right? So I put a note here that the variable importance is not what I expected. And I do not have an explanation to that. But maybe if maybe this algorithm is not identifying the variable that I expected would be the variable that has most importance for the high risk classification, that is the MIC-N expression or the MIC-N amplification. But at the same time, what the variable importance could be telling us is that the classification algorithm of this model is seeing some relationships between the variables that are independent of the MIC-N expression. And so there should be ways where we can have the model increase this accuracy prediction. Yeah, I think 90% is actually very good, is a very good indicator, I would say. because as I said, the one paper that I have in mind said that starting at 70%, a model should be considered good. All right, guys. All right, it's already 8 p.m. We should be good for today, unless you guys have any other questions. Don't
I'm guessing we'll cover variable importance on Monday, because I was kind of interested in seeing um how the variables are used in the predictions, and then also how if there's any point in looking at the outliers, like looking at the cases where the prediction wasn't right, and to see if there's any like relation in between the variable importance and then the cases where the prediction wasn't accurate.
Are you calling the outliers the cases where the prediction is not accurate?
Yeah. Mhm.
We can discuss that, but because of time, we will actually not have time to look into that. If you're interested, I'll be happy to try and help you on answering some of those questions. We can discuss that in the office hours. But the plan for Monday, actually for Monday, the plan is to get us started in the terminal, and we are going to be looking at DNA sequencing methods. As far as the algorithm, as far as this algorithm, this ends here because we were able to use a machine learning method that already gave us a high prediction value. If you really want, we can take a look at the variable importance, how that was done here. We can discuss that on Monday. But as I said, this is not what I expected. I can explain why these variables can be important, but this is not really what I expected. And right now I cannot explain this. That's why I did not want to go over this this section. But we can discuss it a little bit on Monday, if you want, or we can discuss it in an office hour.
Sure. I was kind of just interested more um from like a conceptual perspective, but yeah, I hadn't realized that we were. Yeah, I mean this is already really interesting. I was kind of just interested in like what the typical like next steps would be
Yeah,
for looking at an algorithm like this.
yeah, this is certainly a next step. But I would like us to discuss this in an office hour with the details of the points, and I can really do my best to answer your questions. But because of the time, I think we need to move on to the terminal, which is the plan to get us started on for Monday. Okay. All right, guys, let me just I just have a note here that I need to let you guys know. So my plan was to give you guys a little bit of time for the DNA sequencing methods. I think DNA sequencing is something that is really important, that I want you guys to have that information. That video is already on the YouTube channel. Sorry, the YouTube. I did not put it in the website. I only put it in the YouTube. So I should probably send it to you guys. Yeah, I didn't put it, I did not put a link yet in the website. It's in the YouTube channel. You guys can look for the DNA sequencing methods, because I'll give you some time to watch that video on Monday or next week. But officially on Monday, we are starting the terminal. I'm going to help people do terminal installation, and especially the Windows users on Monday. And I think Etenish and Nithya were discussing holding office hours to help people install terminal in their machines as well. So the plan for next week is terminal. And if you guys have questions about machine learning and the algorithm, I can help you in the office hours. And as I told Eric, I can go through the variable importance part on Monday as well, but I should not spend more than, I don't know, 10 minutes on that. All right, guys, do you have questions or comments? Any other questions or comments? Okay, I'll see you again on Monday. My office hours are in the syllabus. I hope you guys have a great weekend, and see you on Monday, either in the office hour or in class.
Thank you. Bye.
Thank you.
Thank you.
Thank you. Goodbye.
Thank you.